Swayam Prabha. Digital India. Educated India.
Students, welcome back to the developmental biology classes. So in the previous lecture I was introducing and describing about the structure of the female and male gametophytes and how they form, and the main point is that in plants meiosis does not directly make gametes, instead it makes what are called spores. And these haploid cells of the spores undergo mitosis to finally make them gametes. And we were beginning to understand the process of how the male gamete finds its way to the female gamete, and I told you that is the pollination. And to understand pollination, first we try to look at the different kinds of flowers. We saw in some plants both male and female gametes are made on the same plant, and we call that monoecious, and within that you have perfect flower where in the same flower both gametes are made. And then we had staminate and carpellate flowers where the male and female gametes are made respectively. Then we familiarize ourselves with another group of plants called dioecious plants, where one plant makes like one individual member of that species makes only one type of gametes and not the others, and we saw palm trees as an example. Okay, so today we are going to continue on that theme. So there is one interesting phenomena that happens in this pollination process, and that is avoidance of the pollen grain produced by the same genotype fusing with the female gamete of the same genotype. So that is called incompatibility. So incompatibility helps in maximizing the combinations and therefore the genetic diversity generated by the sexual reproduction. So this is achieved by in a number of ways, and there are two broad classes. One is called the interspecific incompatibility, where pollen grain of one species does not fertilize the female gamete of another species, a different species. Then you have something called intraspecific self-incompatibility, where within the species it does not happen. And that is the process that has been well understood, and that is what we are going to look in some detail. This intraspecific self-incompatibility again can be classified into two types. One is called this gametophyte self-incompatibility, and the other one is the sporophyte self-incompatibility. So these two cartoons illustrate this process. So first let us look at the gametophyte self-incompatibility, which is a lot easier to understand. Like for example, the genotype of the plant, the sporophyte that makes this pollen grain, has two alleles of the self-incompatibility locus or the S locus. Let us say it is S1 and S2. These are the two alleles. For a diploid you expect two alleles, and these are the two alleles. So you will have two kinds of pollen. Let us say this is S1 and this is S2. Okay. So now here when for example here S1 pollen landing on the stigma of a carpel in a sporophyte having the S2, S3 genotype. So here if you look at these two alleles are shared. So this also has one allele of S2, this also has one allele of S2. So both this gamete is S1, but this one does not have S1, although they share one allele. So the purely looking at the allele of the type of allele present in the gametes, this S1 does not have match with either one of the two types of genotypes possible for the female gametophyte. It will be either S2 or S3. So, neither or S1. So, therefore, this is a compatible interaction, and you have the pollen grain germinating. Remember, I was telling about in the previous class, while describing the pollen structure, I said pollen has one cell that we call as tube cell and another cell within this tube cell called the generative cell. So, I told you why we call this tube cell will become clearer later, and here you see the tube. Okay. The tube cell, the perfectly sphere like one, germinates and elongates, forming a tube like structure, and this happens when you have compatible interaction. Here S1 and S2 or S3 are compatible, and therefore the germination and therefore pollination is going to be successful. Whereas this pollen with S2 allele for that given self-incompatibility locus shares an allele in the carpel, and as a result this is recognized as self, and when it is recognized as self, this germination does not happen. So, the pollen tube, even if it starts, it degenerates and there is no fertilization that is going to happen. So, this is called gametophyte self-incompatibility, where the gametes sharing the same alleles are recognized as self and pollination fails. Okay. The other one is quite interesting in the sense that although this pollen is S1 and this is on S2, S3, so this is these two alleles are completely different from this, but even then it fails to germinate. That is because I told you that the pollen grain has an outer layer called the exine, and the exine is produced by the sporophytic tissues. So, sporophyte had S2, and therefore the products of S2 allele will be there in the exine, and due to that this plant will, this carpel having the S2 will be able to recognize that component, and as a result it fails to germinate. It is primarily because the sporophytes share a common allele, although gametophyte itself does not. And the mechanism for that is because although this is a gamete having S1 allele, its outer cover is derived from the sporophyte, and that sporophyte has the products of the S2 allele. And this sporophyte incompatibility, therefore this is sporophyte incompatibility, and we call this as such. So this is the difference between the two intraspecific self-incompatibility, where you can't have match between the gametes alleles, then it's gametophytic self-incompatibility, and you can't have match even with the two sporophytes producing the two gametes, and that is a sporophyte incompatibility. So now let us look at mechanistically in a biochemical sense how these things work. There are multiple mechanisms. In some species, for example, the S locus encodes an RNase, and the presence of that RNase itself in the pistil is enough to avoid mating with the same allele coming from the another plant. And there that RNase is responsible for the the failure of the pollination. And there are other species where F-box proteins do the job. And in a third variety, what you have is a calcium wave based rejection. So, when the pollen grain germinates, that is the tube cell elongates and forms this tube like structure, and in this you have a slow moving calcium wave towards the direction of the end of the tube, and this calcium wave is important, and you have calcium channels on the tip as well as on the shanks, and they are responsible for properly guiding and for the successful pollination, for the successful germination of the pollen grain. And in the incompatible situation, it has been observed that this calcium wave is disturbed, and when the calcium wave is disturbed, again you do not have the successful germination of the pollen grain. So this is a third variety. And there is a fourth one which operates mainly in brassica, where it has been extensively studied, and there you have a receptor ligand recognition situation. So for example, when you have a self pollination between the two plants, one S1 S2 and the other one having the same allele, so what happens is the pollen coat contains the ligands, and that binds to so these are cysteine rich proteins, and that's why it's called SCR, and they bind to receptors which are serine threonine kinase. And when you have a compatible, look at the shape of the cartoon here in the receptor, so they are shaped to fit the ligand. So therefore it is compatible interaction in terms of the ligand receptor binding, not in terms of the fertilization itself. And when the ligand fits the receptor, it initiates a reaction, and that leads to abrogation of the pollen tube, and due to that pollen grain, that pollen is rejected. Whereas when the pollen that lands on the stigma is from a different genotype, like this is S1 S2, this is S3 S4, so here you have two shapes to indicate that it is a diploid, and therefore two kinds of ligands are possible, and similarly you can have two kinds of receptors. So the same here, so you have the two kinds of ligands coming, and then you find that their shape does not fit the receptor present in the stigma. So as a result, there is no activation of the downstream signals that leads to the rejection. Okay, so this is a compatible interaction, okay, in terms of the pollen successful pollination. So this is another mechanism by which it works. So in summary, there are different kinds of biochemical mechanisms that helps in the avoidance of pollen grains landing and successfully pollinating the ovules of similar or carrying the same S locus allele. So this is how it looks when you look at the pollen tubes. So first let us look at the control. You can see these long structures. So this is tracing the nuclei of the pollen tube and the generative cell within it. So it's stained for the nuclear DNA and with a blue color dye, and that visualizes the pollen tube that is coming in all of these. You can see them bundle of this, whereas you don't see that here. The pollen tube does not even elongate. Okay, so this is a successful rejection, and this is a successful tube growth initiation. So okay, fine. So that is our discussion on the incompatibility. Now let us continue with the pollination itself. Now once it is compatible and the tube germinates, it starts to grow in some species very rapidly, as fast as 1 centimeter per hour, and in some, like in gymnosperms, it's very slow. It takes longer, like sometimes it can be as long as an year for fully to grow. So this is a scanning electron micrograph showing the style cells here, and this is the pollen tube that's coming down. Okay. And so here you have the nuclei nicely stained with the DNA binding dye DAPI, and you can see the pollen tube cells here, and the elongated structures of the sperm nuclei you can see here. So therefore they grow in angiosperm particularly, they grow very rapidly. And during this process, within the tube cell, you do have the generative cell migrating down. Okay, so this is only one known exception where a plant cell migrates, okay, but not intercellularly, it is intracellularly you have the generative cell migrating. And during this process, generative cell would have undergone mitosis and generated two haploid nuclei. It starts with haploid, so therefore not surprising, you will have two nuclei there, and it will become clearer a little later why there are two nuclei here, how they are going to fertilize, two sperm nuclei. Okay. So this tube generation and its migration all the way going down to the micropyle, as shown in this image, obviously is an elaborate process. So therefore there will be proper guidance, repelling and direction to the right place, all of that will be involved. And experiments so far has shown that the embryo sac itself signals the direction of this pollen tube growth, and experiments have been done where when you have defect in the embryo sac but not in the surrounding sporophytic cells, still there had been problem in proper pollen grain germination, the tube elongation, clearly indicating that the embryo sac does signal the process of tube growth. And in addition, the sporophytic cells of the style itself is also required. For example, it builds up a gradient of GABA, which is a neurotransmitter in animals, and this GABA gradient also matters. There is an enzyme called POP2 that degrades GABA, and as a result it generates a gradient from the source where GABA is produced to the sink where it is being degraded. And the GABA gradient in the style has also been shown to be important for the successful pollen tube growth. And in addition, these two cells, the synergids, they are also responsible in guiding the pollen tube to successfully go to the egg cell. And once it goes there and delivers, it delivers two sperm nuclei. One is going to fuse with this egg cell nucleus, and that is the normal fertilization generating a zygote, and the other one is going to fuse with these two nuclei of the this endosperm precursor cell. This is going to become the endosperm, and this multinucleate cell finally is going to make the food reserve for the developing embryo, which we call as the seed. Okay, so in the seed that you normally see, only a small part is where the embryo is going to be. The bulk of it is actually the endosperm storing the food, and that is what we get from the grains. When you eat rice or wheat, what you are actually eating, the endosperm. Okay. Alright, so that is sort of summarized in another cartoon. So these things we have already gone through. So again have this is the microsporangium having multiple pollen grains developing. So that's shown in this diagram, multiples of them in that, while the megasporangium generates only one. So it's again recapitulated in this cartoon, and you have the two sperm nuclei here, and these are the one is going to fuse with this, another one is going to fuse with this, and as a result fertilization in angiosperms is called a double fertilization. One fertilization is the normal reproductive fertilization, the other one is only going to generate the food reserve. Okay. But interestingly, this fertilization is not really important for embryo formation. There are situations where this haploid cell or even sporophytic cells, need not even be egg cell, are capable of generating an embryo, and that kind of an embryogenesis is called apomixis, meaning no mixing of two different genotypes to make a zygote. and that is agriculturally important, and you can read more about it, but from a developmental perspective it is beyond the scope of this introductory course, so I am not going to get into it. So all you need to know is that in plants fertilization is not essential for embryogenesis in all situations, and there are contexts where the haploid cell can actually make an embryo, or even diploid sporophytic cells can also make an embryo. So the ploidy does not seem to be a crucial thing for embryogenesis. Okay, so that is about fertilization. So now let us continue on that to the next step. Obviously the next step is embryogenesis, how embryo develops. Okay, so we so far saw how the two gametes are formed and how they fuse. So the next, after they have fusion, how the zygote itself develops. Okay, so the embryonic development has three clear purposes that are achieved, and one of them is establishing the body plan. Okay, so we saw that in the Drosophila as well, dorsal ventral axis and anterior posterior axis formation, how that is set up first. And here the body plan involves a radial body plan, like if you take a cross section, from the center to the periphery, so that is the radial plan. So from the periphery you have the dermal, the outer layer of plant tissues, and the cortex, that inner but towards the outer part of the inner side, that ground tissue. Then the innermost, which is the vascular tissues. Okay, so this forms the bulk of the stem or root, and then you have the xylem and phloem, the transporting cells, and that is the vascular tissue. So these are three different kinds of embryonic cells that are originally set apart, something like the ectoderm, endoderm and mesoderm. That is the radial patterning. Then you have top bottom, instead of left to right, here we talk about top bottom because plants are rooted in the soil and they grow away from the soil, and the root goes into the soil, right. So therefore there is an axial patterning as well, that is shoot root. Okay, so here you have three types of tissues and here you have two types, one is the shoot and the other is the root. So these two patterning needs to be established. So that is the basic body plan. And then another important aspect of embryonic development is setting apart meristems. So meristems we will see elaborately a little later. Essentially they are stem cells, undifferentiated cells from which different kinds of organs can form, and they are set apart initially. And most of the plant structures, for example leaves, branches, flowers, everything come from the one of the meristems called the shoot apical meristem, and the root cap etc. form from root apical meristem. So and those two meristems have to be established. And the third, which is unique to plants, is building up lot of food. They are not going to have, like mammals, continuous contact with the mother during the course of embryonic development. Rather once the fertilization is over, they are going to get disconnected from the mother because they have to disperse to different locations. The plants do not walk around, so their embryos have to be broadcast over a big area, which means the product of fertilization, here I want to start using the word seed, has to disperse. So that means wherever it is going to land, to start off its life, it needs to have stored food reserves, and therefore building the food reserve is an important goal of embryogenesis, okay, unlike what we saw in other situations. And so that is the third important purpose achieved during embryonic development. So let us go into each one of them in some detail. So before we go further, since we learned so much about maternal effect genes in Drosophila, to keep the continuity with that, we will briefly think about maternal effect genes as well. So, so in Drosophila we saw maternal effect genes are key. So they set apart the initial morphogen gradient gradients within the zygote, and that drives the rest of the asymmetries all the way leading to segmentation of the body plan. So here, are there maternal effect genes, and do they have that sort of a central contribution? So that's an obvious question. So the answer is yes, they do play a role, but here the situation is a lot more complicated. You just saw in our discussion on incompatibility aspect, the pollen grain has maternal components right around it. So similarly, the embryo sac is deeply inside layers of maternal tissue. Like for example, if you see this, this whole thing is maternal tissue. So this is where fertilization happens. The integuments which are going to form the seed cover. So this is maternal tissue having diploid cells. So due to this intimate association of the maternal tissues, it has been very difficult to really delineate the exact contribution of maternal effect genes. Nevertheless, some of the mutants isolated in Arabidopsis and maize show that the maternal genes do matter, particularly the maternal genes contributing to the embryo sac development. So they have been shown to be important. Okay. So again the same issue, that the embryo develops inside several layers, covered by several layers of the sporophytic tissue. Studying embryogenesis has not been easy. Drosophila lays eggs, C. elegans lays eggs and so on, but you do not have that sort of a thing with the angiosperm, and therefore it's not easy to look at them. So sometimes what scientists do is they look at other plants like primitive ones like brown algae, where the zygote is independent of the other tissues. So there is external fertilization and the zygote is not covered in layers of the mother's tissue. So people have looked at the embryogenesis in brown algae in some detail, and one interesting information that comes from is the role of the secretory vesicles and how the environmental cues impact on the polarity, the anterior posterior polarity, or dorsal ventral polarity that we learnt when we discussed about Drosophila. So here again polarity, the important polarity here is the top to bottom, the vertical axis is important, shoot, root, and there while like in many animals the point of sperm entry initially sets up the polarity, but then that initial polarity cue provided by the sperm entry can be overridden by light, because there is no point if the sperm entry point ends up facing up and lying on the soil, and if you start developing root upwards, it's of no use. So therefore the environmental cues can override the initial polarity cue provided by the sperm entry. So that is an interesting fact of plant reproduction, embryonic development, that came from studying the embryogenesis of brown algae. So here let us look at this series of images. So this is 8 hours after fertilization, immunostained for a protein that is specific to secretory vesicles, and that's what first you start to see. And these secretory vesicles then start kind of moving towards one end, and that is initially set by the point of sperm entry, and that is where the root kind of tissue is going to form, which is called the rhizoid. Okay, so it's here. So in this you can see that asymmetry. So this is forming a structure which we call rhizoid. Okay, and that starts to form, and you can see secretory vesicles, the white color thing, really asymmetrically moving towards this rhizoid end, not to the other side. And these secretory vesicles carry cell wall components that are distinct and unique to the rhizoid, and that is how the rhizoid forming cell is distinguished from the other one, shoot equivalent, called the thallus. And the first cleavage is perpendicular to this axis. You can see here the cell plate forming, and that is how the asymmetry is established. So in summary, the sperm entry site marks the place where rhizoid is going to form, and then you have an asymmetric movement of the secretory vesicles towards the rhizoid, and there they deposit the specific cell wall components that distinguish the root from the shoot. Okay. But this axis formation can be, as I said, totally overridden by the direction of light. So here, for example, this blue color is the DNA of the sperm. So this is the sperm entry point. So initially F actin, which is important for the cytoskeletal rearrangement, gets deposited here, as if this is where rhizoid is going to form, and this is where secretory vesicles are going to come and deposit the cell wall components. But that can be overridden if you shine light in this direction. Okay, so as shown by this arrow. So now that sperm entry point is forgotten, and instead F actin forms a foci focus at the bottom, away from the light at the darkest point. And now the secretory vesicles are going to move here and deposit the the rhizoid specific cell wall components here. Okay, so this is how the apical basal axis. Okay, instead of anterior posterior, here we call apical basal, top bottom axis. And as I said, the distinct cell wall component seems to be key for this. Okay, so now fine, so this is about the asymmetry of the first division we understood by studying brown alga. Similar thing happens in angiosperms as well. So the initial cell division is asymmetric, generating two cells. The small cell is called the terminal cell. This is the one from which all of the embryo is going to form. The other cell called basal cell is not going to contribute to the embryo at all. So this is going to divide and form a structure, a temporary transient structure called the suspensor. Okay. So this is the basic pattern. You have an asymmetric division producing one cell at the top, which is going to form the embryo proper called terminal cell, and then another one called the basal cell, which divides to produce a structure called suspensor. So this basic pattern is common in the embryogenesis of angiosperms, but then among the different flowering plants you have tremendous morphological, that is shape variations on this basic theme. Okay, so that is something we need to remember. That is why all plants don't look like the same. But if you look at mammals, if you go to the very early embryo, as we have learned during von Bayer's last discussion, there is great similarity. So what is the role of the suspensor? So what the suspensor does is, in both flowering and non-flowering plants like gymnosperm and angiosperm, it orients the absorptive surface of the embryo towards the food source. Therefore, it kind of facilitates the embryo to uptake nutrients from the food source. And in flowering plant, it actually acts as a conduit as well for the food to the developing embryo. So that's the role of suspensor, and eventually suspensor is going to degenerate and the rest of the embryo is going to form from this. So the body plan that we talked about, the radial, dermal, ground and vascular tissue, and the shoot and root, apical meristem setting up, they are all going to happen from these cells, okay, embryo proper. So before that, let us look a little about what we have understood about suspensors themselves based on the genetic mutants identified in Arabidopsis. Okay. So that is going to be shown in couple of slides. So this is an overall summary continuing from this step onwards. So the cells here divide again in the I want to reiterate, there are no cell migrations, there isn't anything like gastrulation. As a result, the plane of division, the longitudinal and latitudinal divisions or planes of cell division is really critical in generating the body plan. And after a few rounds of division, you form a structure called globular stage embryo. Okay, and in the globular stage itself, as we will see in couple of minutes, the radial patterning gets established. And from this, when the cotyledons, the seed leaves start to form, so these projections, these mound like thing forming, this globular then ends up changing shape into heart shape. So this is globular embryo, this is heart embryo. And in some cases where this develops really elaborately and to be fit within the seed cover, remember the integument is going to form the seed cover, to package it when it bends like this, it forms a torpedo like shape, and therefore it is called torpedo stage. Okay. And during this stage, clearly the apical and basal polarity becomes very apparent once this mounds of cells, that is the two cotyledons, start to form. This is in dicots. In monocots you have only one cotyledon developing. So here you have the root meristem and here is the shoot meristem. Okay, and these are the two cotyledons. So this is an overview. So we will get into some at some detail each of one of these. So first let us look at the suspensors. So this is the micrograph of an and now wild type embryo. So this is the suspensor attached to the via the placenta to the sporophytic tissue, and this is the embryo developing. So in a mutant, in a gene called sus1, which is called dicer like 1 because of its similarity to the dicer. Dicer you may have heard in discussions on RNAi. Okay, so dicer is a protein involved in cleaving RNA into small signaling RNA molecules, so called siRNAs. They are involved in even in miRNA generation. So miRNA are usually transcribed as tandem repeats, and then each of the repeat has to be cut into the monomers, and that's done by dicer. Okay, so it's basically an RNA cleaving enzyme. And sus1 bears similarity to that, and as a result it is also called DCL1. And mutations in that protein results in embryo-like structures forming out of the suspensor cells, indicating that in the wild type sus1 is important to prevent embryo-like development, which further indicates that suspensor cells have the ability to form embryo. And based on this and other mutant studies, the current model of how the embryo suspensor polarity is maintained is explained in this model. So you have signals coming from the embryo telling the suspensor not to form embryo. Okay, so it's a suppressor. So suppressor is usually indicated by a line like this. This sort of a structure is suppression in developmental biology, and this arrow is an activation. Okay, so signal from the embryo suppresses embryonic development in suspensor cells, and signals from the suspensor cells promote embryogenesis in the embryo. So this is what currently scientists think the way signaling happens between these two to ensure suspensor remains a suspensor and allowing embryo to develop normally. Okay, so now let us continue with the establishment of the body patterning. I told you in globular stage itself the radial patterning becomes obvious. That is indicated in this color coded cartoon here. So the outer is the protoderm. So this is the one that is going to make the dermal tissue, which is going to be the outer cover of the plant organs. and then you have the inner tissue from which cortex, pith, etc. are going to form. The central portion of the, not the very central, but between the, the vascular tissue giving cells and the outer periphery, that is the dermal tissue, the in between layer, that is the ground layer, and that is this ground tissue. Then you have the procambium, which gives rise to the vascular tissues, that is xylem and phloem, for conducting water and food respectively. Okay, so this becomes clearer at globular stage itself. And once the cotyledons are going to form, cotyledons do not form only from the protoderm, okay, it is derived from the general embryonic cells. And when that is going to form, you get to the heart shape, and I just told you that by the time the apical basal polarity becomes obvious, and that is indicated in this cartoon by showing where the apical shoot apical meristem is set apart and then the root apical meristem is set up. Okay, so these two gives you that by this stage the apical basal polarity has been already established. And in this process, the plant hormone auxin plays a important role. Again, I want to highlight before I get into auxin, in the absence of cell migration, regulation of the planes of cell division, longitudinal and latitudinal divisions is really critical, along with cell expansion for the proper morphogenesis during embryonic development in plants. Okay, so there are no cell migration, so therefore planes of cell division and cell expansion are important, and only by changing these two, all the different shapes are generated. That thought itself is quite fascinating. Okay, without migration, simply by changing shape, changing the direction of expansion and plane of cell division, you can create a variety of shapes. Okay, so now let us move to the auxin discussion. So initially, auxin moves, like for example in this 4-cell stage, it moves apically, and then later it moves towards the hypophysis. That is the junction between the embryo proper and the suspensor. Okay, so that is where the root meristem is going to be set apart, and it starts moving towards this. And for this, a key protein that is required is this pin family auxin efflux carrier. So these are important for carrying the auxins in the direction shown. So they are produced asymmetrically, and that asymmetrical expression is key for the direction of auxin movement, initially to apical portion and later to this hypophysis site. Okay, and the pin family localization, rather their expression itself, is controlled by a protein kinase called the PID1, and that is very critical for the directionality of auxin movement, and mutations in that affect proper setting up of the root meristem. Okay, so we will see that as we move on. So here, for example, in this wild type, so based on this kinase, PID kinase, sorry, I said PID1, so it is actually PIN1 and it is just simply PID. So the PID protein kinase is responsible for the normal PIN1 localization. So based on the difference in expression of these two, PIN1 gets highly concentrated here, and as a result auxin flows to this, and that auxin is responsible for setting up the place where shoot is going to develop. For the shoot apical meristem formation, flow of auxin towards this place is important, and that is made possible by the high concentration of PIN1 in this region. So now if you overexpress PID1 as shown, PID in the apical part, now pin 1 doesn't get localized here, its expression is not concentrated here, and as a result auxin doesn't flow towards this. So what is the consequence? So if you look at the wild type, due to pin 1 localization here, auxin flowing there induces the auxin induced gene expression in these cells, okay, and that is critical for the root development. On the other hand, when you don't have that pin 1 concentration there, auxin flows everywhere, and as a result these cells start expressing auxin target genes, that is auxin induced gene expression, and due to that you have everywhere root like structure forming, and that abrogates the normal embryo development, okay. So the last part, in the next 2 minutes I will just explain how auxin works, okay. So the understanding of how auxin works really helped in connecting plant hormones to the downstream gene expression and the actual cellular events leading to development. So auxin is indole acetic acid. You can see here, if you remember tryptophan, that has the same side chain. So this acetic acid having indole, so it is called indole acetic acid or IAA. So that's why this abbreviation IAA comes. So normally in the promoter region of auxin target genes, you have this auxin responsive factor ARF bound, and this doesn't bind all by itself, it also has another protein bound to it which is called AUX protein or IAA protein. And when they are bound like this, ARF is kept inactive and the downstream gene expression is not activated. But if auxin is present, auxin binds to this SCF, which is actually an ubiquitin adding enzyme, and that ubiquitinates this, which is shown in the next step. So in the presence of auxin, auxin binds to SCF and that activates it, and that ubiquitinates this AUX protein, and once ubiquitinated it is targeted to proteasome and gets degraded. And freed by this auxin, now ARF becomes active and that induces the downstream target genes. So this is how the auxin signaling works. So essentially normally the targets are kept suppressed by a suppressor binding to the auxin response factor, and in the presence of auxin that suppressor gets degraded and therefore the suppression is removed and the gene expression is activated. So this is how the auxin expression, auxin induced gene expression is activated, and many mutant analyses have shown that this process is key for the normal formation of the root structures here, okay. So with this we will stop today, and then we will continue with the embryogenesis, the next steps, that is setting apart the apical and basal, the two meristems, like shoot apical meristem and root apical meristem, and food reserves in the next class.